Yeah. Okay, let's go ahead and start. The lockdown round.
That was very hard. I was called scared. I think I was just getting trying to get.
If you had an issue with the lockdown browser and wouldn't let you take your test, let me know.
It could have been.
You did?
Let me need it. I don't know how.
I can't do. One of the things that you can do is reset exams and remove lockdown browsers if necessary. So if you had an issue and you couldn't, and wouldn't let you take your test, and you're like, oh, I'm running out of time, I really must take my test. Well, life, have an issue with it, let me know. Hopefully we want them to back to the right test class. So, what is the hurricane? Okay, let's for today and hopefully start capital metabolism. The last thing we were talking about was these tumor suppressor genes that protect us. And the tumor suppressor genes prevent us from getting cancer developing. Now that doesn't mean we're never going to have cancer because of the tumor suppressor genes, but these genes are sometimes genetically programmed to fail for different cancers. They are, when we say that the cancer runs in the family, well, it's probably because their P53 gene in their DNA that would protect them for, let's say, colon cancer. Hey, it wears out and then it fails. And so you're the individual with that particular P53 gene is more prone to colon cancer. It doesn't mean they're going to get colon cancer. It's all it means is they're at a higher risk now. So if you have a history of colon cancer in your family, it's likely because of the tumor suppressor gene failing to do its job. But we don't know what's going to fail. We don't have any idea why certain genes, the tumor suppressor genes, the P53, what part of it's going to fail. Why do you get breast cancer? Why do you get colon cancer? Why do males get prostate cancer? You know, we're exposed to all the same types of insults from the world around us, from the food we eat, radiation, all these other things we see, and yet many people go their whole lives. They're 95 years old, and you know they're going just fine. And someone that's 40 years old gets prostate cancer or breast cancer or or colon cancer. We don't know why the gene is destined to fail. Okay. Now, however, what the tumor suppressor gene does for us, though, is if there is damage to the DNA, and that's where a cancer cell thrives. Cancer cells, not cells, will alter the DNA of a healthy cell so the cell goes into mass production. Because cancer is nothing more than uncontrolled cell growth. And so when we have uncontrolled cell growth, we have an abnormal number of cells crowding into the same place. They all look irregular. I'll get it. They don't look normal. They have unusual shapes and sizes, and they're piled up on top of each other. And what happened? What will happen is that these cells will continue to grow and spread across the body, and then eventually it'll kill the host. Because cancer cells, that's what the malignancy does. It thrives and takes over grows and grows in the body and sends out seeds for more tumors, and tumors will grow, multiply, and eventually will impact major organ systems and cause death. What the tumor suppression, that's what a cheery topic for one for the first thing Monday morning, isn't it? We're all gonna die from cancer. Well, not necessarily. Probably not. What the tumor suppressor gene does though, is it detects a problem with the DNA. It will stop the cell cycle in the S phase. Remember, we have G1 is the growth phase, S is the synthesis phase where we copy our DNA so we can make daughter cells that are identical to the parent cell. During the S stage, if there's a problem, we identify the problem right at the beginning of the S stage, if there is a problem. If there is, then P53 can take over and can repair the damage. And it will activate enzymes that can slide up and down the DNA strands, chopping out the mistakes. Because that's what a mutation is. A mutation is just a mistake in the sequence of the bases. Typically we would pair adenine to thymine on the DNA strand, or cytosine to guanine. But sometimes we pair cytosine to cytosine, or thymine to guanine, or something that's not supposed to be there. And if that is copied into the daughter cells, then that mutation, that mistake continues to spread. So what we'll do is we'll stop the cell cycle. P53 gene will stop the cell cycle, initiate repairs, and then restart the cycle if everything's fixed. So and of course, we're never aware of this because we're talking about something that happens. The entire copying of our DNA takes about 2 hours out of, a lifetime, a lifespan of maybe 4 days. So we don't really notice it because we're still running an interphase the whole time. We're just replicating the DNA just a few minutes before we go into mitosis. So we fix everything and we're good. And if we can't fix everything, then there's 2 options. The cell can initiate, the tumor suppressor gene can initiate apoptosis, which is cell self-destruction, or it can put us into senescence, where the cell doesn't replicate any further, the whole thing stops, and eventually the cell degenerates. So either way, the tumor suppressor gene, when it's doing its job, is protecting us by either fixing the damage or eliminating the problem. And I mentioned the other day that cells that are very close to, that have very short telomeres, can easily be put into senescence. Remember what a telomere is? Telomere is the structure on the end, and they took all my markers. Telomere is like on a shoelace. This sounds silly, but as an analogy, it works. On a shoelace, those shoelaces have these little plastic caps on the end of the shoelace, so you can get it through the eyelet in your shoe. That's what a telomere is. Telomere is just this top portion of DNA. And here's a strand of DNA right here. And up here are a series of nucleotides that make up the telomere. Well, the theory is that as every time a cell undergoes replication, we chop off a little more of the telomere, and as it gets shorter and shorter, the cells are less likely to undergo mitosis. Well, cells that are close to the end of their telomere are more likely to go into senescence than they are to be destroyed. Either way, they're not going to do anything. But the conventional thinking is that the telomere helps to determine the age of the cell, of a cell line. Every time a cell is replicating, it's part of a cell line. So as we get shorter and shorter, that line runs out. Typically, it's sort of thought there's like 50 replications in a line before it stops copying. So since we have 1 trillion cells or more in our bodies, we don't notice. So cancer cells play dirty though. Cancer cells don't chop off their telomeres. Cancer cells keeps the telomeres, that's so, I know you can't read it, but that says telomeres. I'll just save the question now. Cancer cells don't have their telomeres chopped off. They can keep copying and copying and copying as they go along, and their cell lines never wear out. So, anyway. Now, we've all experienced this. You know, consider, I mentioned this the other day, this is a really important point, so I'll say it again. Ultraviolet radiation is probably one of the easiest ways for us to damage our DNA in our skin cells. Too much sunlight. Sunlight contains ultraviolet radiation. We need that UV light to stimulate, among other things, generation of vitamin D in our skin. Vitamin D is what we use to help absorb calcium, which we need for our bones. So everything's sort of all linked together here. If a person is exposed to too much ultraviolet radiation, and depending on the pigmentation of their skin, it may be significantly noticeable or not, doesn't matter. There's going to be a massive die-off of skin cells, because it's going to kill the healthy cells. It's going to damage the DNA of the healthy cells in the dermis. Ultraviolet radiation penetrates through the epidermis into the dermis. It damages the DNA of the cells in there, and the P53 response is everybody dies. And we see healing of patients. Everybody can be burned, regardless of skin color, and everybody will heal again, regardless of skin color. The marker, the indicator, that's an indicator that your P53 gene is doing its job, because it's killing off all those healthy cells that became damaged, and so they all rise to the surface at once. Because you never see skin cells coming off individually. Oh, there goes one right now. We never see that. The only time we ever notice is we have large patches of skin coming off, dead skin, thanks to the P53 gene. So, can't emphasize that enough, how the P53 gene is going to be protecting us. Okay, now moving on. Protein synthesis. This is what DNA is all about. The DNA is a gigantic cookbook for it. It is a cookbook made up of strands of DNA that's 6 feet long, microscopically thin. The order of the bases that make up the DNA are to provide the order of the amino acids that make up a protein. Remember, proteins are made up of amino acids. You have amino acid 1 plus amino acid 3 plus amino acid 19 plus amino acid 2. That specific order means something to a protein. That's the order of only 20 amino acids. The order of the amino acids dictates what the protein is. So you may be using, lysine or galactose or leucine or valine or glutamic acid. Those are all amino acids, and they all do different things. And the order of the amino acids dictates what the protein is going to be. Question? We're just holding your hand up.
Sorry.
No, don't apologize. Okay. Now, how the DNA does that? The DNA provides the information on how to make these proteins. The DNA uses the order of the bases in the DNA strand. Here's the strand right here, and you would have, adenine, and you would have cytosine, and you'd have cytosine, you'd have guanine, you'd have thymine, you'd have another adenine. That means something. The order of these bases codes for an amino acid. Order of bases codes for amino acids. Everybody read that? Now, so what that means, feel better. Thank you. What that means is that we now have a cookbook, we have a recipe, we have a way to direct the formation of all the proteins our bodies need simply by having it stored in the DNA. You have a cookbook. Cookbook is contains recipes. You go into the cookbook, you got grandma's cookbook, and there's your recipe for homemade biscuits. Change. Maybe grandma made caramel biscuits or whatever. So, and it gives you all the step by step sequence. It tells you the eggs, the milk, the butter, the flour, whatever, the salt that goes into the shortening. Whatever goes in the biscuits. I have no idea what goes in biscuits. But it tells you the sequence because the sequence is important. And the sequence is important in baking. Sequence is important in producing a protein. You have to do things in the right order. You know, you don't put the bowl in the oven at 350 before you've added the ingredients, right? I mean, I might do something like that, you know, but you know, the sequence you call it, you break the eggs before you put them in the bowl. You just don't throw them in, okay? The sequence of the instructions in a cookbook relates directly to the sequence of the bases in the DNA. And so every gene when we talk about our genes are nothing more than the physical location on the DNA strand for a set of instructions to make a unique protein. And this set of instructions is based on based on the order of the adenine, guanine, cytosine, and thymine on the strand. The sequence of those bases dictates the amino acids that are going to be linked together to give you a unique protein, whether it's hemoglobin or hair color or eye color or an enzyme, catalase, oxidase, any of these, amylase, salivary amylase in our mouth, which starts the process of digestion. As soon as we eat starches, we start digesting foods, starches in our mouth because of the presence of this enzyme amylase. That's a protein. And so we make all these proteins in the cell following the instructions of the DNA. And every living organism does this exactly the same way, whether we are a bacteria or a plant or a goldfish or a human. We all do it exactly the same way. So the genes are the information that we need to make the protein. The genes are made up of a specialized sequence of the bases. Now within the gene, there are sections that are coding and non-coding. Parts of the DNA, this may be a coding section, and this might be non-coding up here, or maybe down here. Non-coding. We don't know what the non-coding regions do, but they're there. They have some purpose. We call the area that is the coding section the exons. Nothing to do with gasoline. The exons are the coding regions within the gene. The introns are the non-coding area. There are areas that are, I guess, maybe they're filler, maybe they're spacers, maybe they have a purpose, maybe they're just places to hold on to more bases. We don't know, but the non-coding regions are interspersed among the coding regions. So the exons are the important portion, and the introns are we'll just say they're non-coding. Because 10 years ago, we used to call them junk DNA. We don't call them that anymore, because there are purposes for them. We just are not certain exactly what they do, but there is a reason for them to be there. So the gene is made up of exons and introns. The code, as I just said, is made up by the specific order of the 4 bases: adenine, guanine, cytosine, and thymine. The code is designed to work in triplet form. What that means is if you have a strand of DNA bases are going to be organized in clusters of 3. So you would have, here's a DNA strand here. Adenine, thymine, cytosine, guanine, cytosine, cytosine, adenine, thymine, thymine, guanine. This is a triplet. This is another triplet. Right here. The triplet, the bases are organized into triplets, and the triplets code for the amino acids. For example, if you have guanine, cytosine, it's going to code for the amino acid proline. If you were to take guanine, cytosine, that's going to code for arginine. So each base, each triplet of 3 bases is going to code for a unique amino acid. So it makes it a little bit easier when we're copying our DNA, when a gene's making a protein, we don't have to get we have to get every base correct, but they come in triplets now. It's a little easier to manipulate because they're in triplets. So we have this three base triplet as the code. We okay so far? Question?
What is the second one say under BCC dash?
Oh, that's arginine. Let me write it out. It's ARGENE.
What's the top one?
Proline. There we go. How's that? Is that better? I have ordered markers that have the real wide tips so I can write in bigger size and maybe it'll be clearer. I doubt it, but it'll still be a scribble. Okay. The triplet codes for a particular amino acid. So that makes it, so now we have a combination of 3 for an amino acid. We only have 20 amino acids. So we're going to make up these combinations. So we have unique combinations for every amino acid. Let's see if this works now.
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has 1 more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidine. For convenience, the carbon atom in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Nucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of the nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain, DNA, on the other hand, consists of two polynucleotide chains. The two DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Okay, so DNA is made up of these four bases joined together with complementary, they pair up in complementary bonds. Cytosine to guanine, adenine to thymine, or adenine to thymine and guanine to cytosine. Those are complementary pairings. You see hydrogen bonds that hold those bases together that causes the whole molecule to twist, and that's what gives us the double helix structure. It's a very simple structure. It took years to figure it out. But the structure itself is held in place simply by these little weak hydrogen bonds right here, which is good for us because if we want to open up the strand to copy a gene, we can. Now remember, the genes occur on either side of the strand. Because when we when we copy the DNA in the S phase cell cycle, we open up the strand. Everybody remembers that? Thank you. So, the strand opens up and we make a complementary copy of each strand, but the genes are located on either side of the strand. So whether we have the top, if we have, here's a strand and here's a strand up here, but the gene could be up here, the gene could be down here. We take advantage of both sides of the strand. It isn't both layers make up the gene on each side, probably doing different things, since they're gonna have to be the complement of each other. So so it really isn't that important to remember that they are purines and pyrimidines. I don't think anybody's ever going to ask you that question. What you need to remember is that in DNA, it's only made up of four bases, and thymine always pairs to adenine in DNA, and guanine always pairs to cytosine. Uracil only shows up in RNA. There is no thymine in RNA. You'll see this again and again here. Thymine doesn't occur in the RNA. RNA is a single strand, and we substitute uracil for thymine there. Questions on any of this? This video, the animation can be tedious, but it does make some good points. Okay. So what is RNA? Well, we know it's a single strand. It's made up of adenine, guanine, cytosine, and uracil. So it doesn't have thymine in here. It is made in the nucleus of the cell itself, in a structure called the nucleus. Inside the nucleus, if this is a cell, here's our nucleus right here. And there's usually a dark spot in here called the nucleolus, and that's where RNA is manufactured. Now, and so they're all assembled from nucleotides. Every nucleotide has a 5-carbon sugar, a phosphate, and a base. In the RNA, the bases are going to be adenine, guanine, cytosine, and uracil. In the DNA, the base is going to be adenine, guanine, cytosine, and thymine. That's what a nucleotide is: a phosphate, a 5-carbon sugar, and one of the 4 or the 5 bases that we encounter. We've got 3 types of RNA. We got messenger RNA. Messenger RNA is going to get made in the is going to be copied off of the DNA strand itself. We have ribosomal RNA, which makes up the ribosomes, and we have transfer RNA, which probably has the most important job. Transfer RNA has to bring the amino acids to the assembly point so they can start linking the amino acids together to make a protein. Messenger RNA is a single strand. It's copied off of one side of the DNA. The DNA opens up. DNA will open up. It'll split in half. Probably the same enzyme helicase that opens it up to make a copy of the DNA. A strand right here. Adenine to thymine, cytosine to guanine, cytosine to guanine, thymine to adenine, and so on. What we'll do is if we open up the strand like this, and down here we have cytosine, adenine, thymine, and guanine, we will assemble a messenger RNA strand off of this. Guanine, uracil, adenine, cytosine. This is mRNA. It is copied off the DNA. It is a complementary copy. So if the DNA strand had cytosine, the RNA strand is going to have guanine. We maintain the triplet sequence. So we had, here's a triplet right there. There's a DNA triplet there. Here we have an RNA triplet right here. Remember, this is the complement to the original DNA strand. We call in to avoid confusion, we call the RNA triplet a codon. RNA triplet. Remember, the triplets are going to be complementary to the DNA triplets. That's all they are. We okay so far? Yes, no? Okay, now. Which way? Okay. That is messenger RNA. Messenger RNA is a copy of the DNA strand. We call it transcription. We are writing down the instructions. We're taking the instructions from the gene and we are essentially writing them down onto a messenger RNA strand. So the gene's saying, here is the order of the bases to make the protein hemoglobin. Everybody knows what hemoglobin is. And so here's the sequence of the bases to make hemoglobin. The messenger RNA starts copying this down. Only they make a complementary copy. So the DNA says we start with cytosine, guanine, and adenine. If we start with cytosine, guanine, and adenine, the messenger RNA strand is going to be guanine, cytosine, and uracil. In the complement, remember the complement in RNA is adenine to uracil, cytosine to guanine. So we make a complementary copy on our RNA triplet. It's a copy of the DNA. It's just a complementary copy. Everybody good with that? Everybody happy? Are you happy? Sort of.
They like me.
That's fair enough. So, okay. Are you still on it?
Yeah,
Okay.
I'll get there.
Now, ribosomal RNA is easy. Ribosomal RNA makes the ribosome. With the ribosome, the site of, we said that's one of the organelles, it is the site of protein synthesis. It is not in the nucleus, it's out in the cytoplasm. The ribosomes are stuck to the rough endoplasmic reticulum. It's what makes the rough endoplasmic reticulum rough. The little dots are all studded on the surface of the rough ER tubules. And those little dots are ribosomes. The ribosomes look like a big hamburger bun with the big bun part on top and the base underneath, and it comes together into two parts, and it's made up of ribosomal RNA. And as soon as it's done its job, it breaks apart and disintegrates back to its nucleotides, and then it gets reassembled. So nothing ever, nothing, everything is recycled again and again and again. The transfer RNA carries the amino acids. Now, the amino acids are found in the cell. We make about 12 amino acids. 8 of the other 8 amino acids we get from our diet. They are present in the cell. And so what the transfer RNA will do is it will bind to a unique amino acid. And it will bring the transfer RNA has a sequence of bases along its bottom called the anticodon, which is the complement to the codon. So if the codon on messenger RNA we have a section of messenger RNA right here, messenger RNA. Let's just say our codon is CCC. This is messenger RNA. So it's the sequence is the codon, which is the triplet in RNA world. The codon, it was the DNA. In the DNA, the triplet was G, G, G. Well, there's our, y'all agree that C is the complement to G? C, okay, this is the complement, they are the complements. There's our codon here in RNA. The anticodon, which is attached to the transfer RNA, is exactly what it sounds like. Here is the transfer RNA. Here, it has its amino acid up here, and down at the bottom of it is called the anticodon. Anticodon. Making it worse, but that's okay. Anticodon. And the anticodon is going to have the sequence of G, G, G. If what's the complement cytosine? 20. And so the complementary, the anticodon on the transfer RNA has to have 20. Yes.
Is that AA on top?
Where?
That-
Okay, that says amino acids. Let's just say it's amino acid equal 1. Sorry. It did say AA, but it should have said AA1. So our transfer RNA is not random. Transfer RNA that's bringing the amino acid in place is following the instructions found on the messenger RNA. So if there's a codon on the messenger RNA that says CCC, the anticodon is going to be GGG. If it would be CGA on the codon, CGA, then the anticodon would be GCU. It would be the opposite. It's the complement to it. So let's see if we can make it any worse. So the whole thing, the summary, the real brief summary protein synthesis has two stages. It has transcription, where we copy the information off the gene in the DNA onto messenger RNA. That's where you're writing the instructions down. Grandma has this great recipe for blueberry pie that she never lets out of her house. She won't let you take it upstairs and copy it on her printer. She makes you write it down, word for word, right in front of her. Because there's all sorts of little subtle things that she adds in. You know, pinch this and dash that, whatever those instructions mean. That is transcription, that is writing it down. So you are copying the information in transcription from the DNA onto the messenger RNA. Whatever sequence of bases you have on the DNA is copied onto the messenger RNA as a complementary copy for C to G, A to U in RNA. Now, we've got the instructions. Now we have to make the protein. That's called translation. You take the recipe for blueberry pie, and now you follow the instructions in order to make the finished product, the pie. Well, here, the finished product is going to be the protein. So the translation is taking the instructions and putting the amino acids together to give you that particular protein. So transcription takes place in the nucleus. Translation takes place in the ribosome, out in the cytoplasm, or rather out in the rough ER. So we copy the information onto a messenger RNA strand. We copy it exactly as it's presented on the DNA with all the exons and introns intact. We'll have to cut out the introns later on and put everything back together. But right now we make a complete copy. Transcription. We open up a strand of DNA where the gene we want to copy is located. Whatever we want to copy. There are millions and millions of genes and gene locations on a DNA strand. We just don't have one place only for a gene. We have lots and lots of copies of the same gene. So we open up the strand right here, where our gene that we want is here. There might be another gene down here for something else. This may be hemoglobin, this may be hair. They are still the sequence. So we open up the strand, and then we use this enzyme called DNA polymerase. I'm sorry, RNA polymerase is the enzyme assembles the messenger RNA. We start bringing in nucleotides. Here is an adenine this is the DNA strand here. Here we have adenine, cytosine, guanine, thymine. On the messenger RNA, the RNA polymerase are going to start assembling a strand of uracil, guanine, cytosine, adenine. Nucleotides are all the same. Nucleotides are either going to be a ribose sugar, a phosphate, and one of the 5 bases. You're either going to have adenine, guanine, cytosine, thymine. If you're an RNA, you'll have uracil, you won't have thymine. So the nucleotides all look the same, except for the base. And so we get the instructions, we're copying, there's the gene we want to copy. We separate the DNA strands. This is the other side of the strand. We're not worried about that right now. And so we are assembling a messenger RNA that is the complement to what's on the DNA strand. We have a start point and a stop point on here. The start, it tells us where to start. So we start here. here. We stop. But there's another gene starting up here, and there's another gene over here. We only copy the section that we want to copy. If we are a little low in hemoglobin, we search out all the genes for hemoglobin in our DNA, probably thousands of them. We don't just have one location. And so every DNA strand is sitting there with another gene next to it on either side of it. We isolate the gene for hemoglobin and we start making a copy of it. We use the RNA polymerase to assemble the messenger RNA. And when we're done, we let it go and it, leaves the nucleus.
As our hair color changes with age, are we creating a new strand of like the messenger RNA that changes that hair color? We were talking about it showing up in the gene.
It can. What will happen too is as our hair gradually loses its color, we're making less and less of the protein. So the gene is no longer effective in making that, or it does change. We go from, we may have bright blonde hair as a child to brown hair as an adult, or the other way around. The DNA, depending on how the DNA changes over time, because it can change, and maybe we are not express, maybe some of the bases aren't functioning anymore, and that's why our hair comes out darker or lighter. And then when we stop making the protein, that's when we start having these transparent hair follicles. Okay, so good questions. Thank you. I've never been asked that.
Well, I was thinking like, is it pulling on like a different part of our genetic code? Like if we go from blonde to brown, if we change like where we pull that piece?
And that's also one too. Maybe the dark hair is a recessive trait, the light hair is a dominant trait, and as that dominant trait wears out, the dark hair goes on to the next trait. So it's like eye color changing over time.
Actually, how we change in the age, you know, I was thinking maybe that was.
Exactly. We're still getting effective pigmentation. It's just different. Okay. Now what you're seeing here, this is transcription. Let's see if I can start this over again. Okay, let's stop this. Okay, so what you're going to see here is the assembly of a messenger RNA strand off of the DNA. The DNA splits apart. The gene is on the bottom strand. So the complement is all the way, the complementary copy of the bottom strand is what's being assembled. We see the DNA separate. In red is the messenger RNA. I know it because it's a uracil. But this is the DNA strand. This is both, these are the triplets here. We're starting to assemble a complementary copy on this side only. Doesn't matter which side, the side that has the gene is the side we're trying to copy. So we'll pair adenine to thymine, uracil to adenine, guanine to cytosine, cytosine to guanine, and so on. This will be complementary, but wherever the start sequence is, we start here. There's a stop sequence somewhere out there, and we will continue to assemble the messenger RNA till we hit stop. There's always a stop sign at the end of the gene. We can start sign and a stop sign at the beginning and the end of the gene, and then that way we don't get any, we don't copy anything upstream or downstream. We start and stop at the right location. So we have initiation, where we open up the strands with the RNA polymerase. We start bringing them together, bringing the nucleotide, the RNA nucleotides together in sequence. That is the elongation, where it gets longer and longer and longer. And then we stop at termination. And then we we now have a strand of messenger RNA that is a copy of the sequence it's a complementary copy of the sequence of bases in the gene. So, because You gotta remember, this is not the DNA strand here in red. This is the complementary copy to it. So if the DNA strand is say guanine, the copy will have cytosine. So it is the complement, it is the opposite pair. It's like adding all these pairs to uracil in RNA. So if you have adenine on the DNA, you're gonna have uracil on the RNA. It's still the sequence, though. It's still the sequence. And if you think about it for a second and try not to overthink this. The sequence on the messenger RNA here is the exact sequence on the other side of the DNA strand. So you are still getting the original strand. You're still getting the original information. So. Okay. Now, there is a problem, because we copied all the introns, we copied all the sections of DNA of the DNA that are not coding for something. We don't know what they are. So we had this really cool little enzyme called spliceosomes. I mean, this is like, really? Yeah, that's as technical as you can come up with. A spliceosome cuts out the introns and exons and then assembles the exons into one complete strand. I just think it's a cool. I'm using my spliceosome today. So we cut out the introns and they go off. We can still recycle those nucleotides. We don't really know what happens to the other, why the other DNA is there. And then we glue, where literally, that's the spliceosome. So they cut out the introns and they glue all the exons together. Now we have a shorter strand that is correct. And we do that we don't do it to the DNA, we do it to the messenger RNA. So we copy the whole section of the gene, then we cut out the non-coding material, at least what we don't need to make the protein. So now our strand is shorter. We have processed the messenger RNA. We are now ready to leave the nucleus and go out to the ribosome. And that gets to translation. That's where we actually take the information and we use it to start assembling, to assemble a protein. So when we say that DNA is our genetic code, it tells us it's a cookbook. It tells us everything about us. It has all the protein information for all the proteins we need. It's true. Everything that we need for our bodies is arrayed in the DNA. Instructions on bone assembly, blood cell making, hemoglobin making, hair color, eye color, skin color, every enzyme we want. All the proteins, the contractile proteins in our skeletal muscle, that we can make our muscle cells larger. Can't copy the muscle cells, but we can make them bigger and more and stronger, more powerful by laying down more and more protein, contractile protein. All those instructions are contained in our DNA. The instructions on how big our bones are going to be when we hit puberty, we grow to our adult stature. You know, the sequence of how many proteins we have to make our bones big. The instructions that will tell us to keep remodeling our bones from the time we stop growing in stature at about age 23 to the day we die. We are constantly remodeling our bones, and that's controlled by the DNA too. The sections of the DNA determine the proteins that are produced there and determine what we're going to do. They're going to determine, how thick is our bone going to get? How thin is our bone going to get? How tall are we going to be? So, and that's what the DNA controls. And since the DNA can never leave the nucleus, we would not want the DNA to leave the nucleus. We don't want it to get corrupted. We never let DNA out of the cell's nucleus, so we use the copy of it. The messenger RNA, it is the transcribed copy, and that goes out to the ribosome with the instructions on how to make a specific protein. And since there are millions and millions of ribosomes in every cell, we are constantly making proteins all the time from all these little ribosomes. They come together, they make a protein, they break apart, they come together, they make a protein, they come together again, and they keep doing that, but they don't stay there. So. Now our genetic code, DNA is made up in triplets. The triplet consists of 3 bases, and we only have 4 bases all together, adenine, guanine, cytosine, and thymine. If you calculate out every possible combination of triplets, I'm not going to do the math. There are 64 possible. Codons that you can have. So you have 4 bases. You can see what it says there are 4 bases and three possible combinations. So it works out to 4 cubed, which is 64. So take my word for it. I'm taking their word for it. But 64, there are 64 possible combinations for the amino acids. There's only 20 amino acids. We have 64 possible codons. We have a lot of redundancy. There are many codons that copy for the exact same amino acid. So some of the amino acids that are really important to us, we have lots of copies of the codon. So that even if we make a mistake, we can correct. Maybe the mistake is still gives us the same amino acid. Yes.
So uracil and thymine is considered one, basically, because that's you said thymine, but on there it says uracil.
Right. And then you only put the DNA. So we're looking at this is the codon, the triplet code for the RNA. So okay, yeah, that's a very good point. Thymine is in DNA only. It's replaced in RNA by uracil. So when we look at our triplet code here for RNA, the codon, we look at adenine, guanine, cytosine, and uracil. So the DNA still has its thymine. For whatever reason, the messenger RNA doesn't use thymine, it uses uracil. I guess it's just to confuse anatomy students. I'm trying not to confuse. So we got 64 possible codons. We have 3 stop codons that tell us that's enough. Stop. Tells us to stop the process of translation. Because there may be another strand coming into the ribosome. There shouldn't be. The ribosome should break apart and form new. But sometimes mistakes happen, and another strand of RNA, messenger RNA, should not be going in. But if it does, there's still a stop codon to keep us going any further. The reason that there are so many possible combinations is redundancy. We don't want to make mistakes. And if we do make a mistake, maybe the mistake still works. So for example, let me show you this sequence here. This wins the prize for the most confusing slide that I'll ever show you. However, if you look at it, it contains every possible codon that we would encounter. You know, all these are codons: adenine, cytosine, uracil, cytosine, guanine. Now, to figure out what amino acid is coded by a specific codon, all we do is I'll just make up, how about this? How about CCU? Okay, this is the first base, this is the second base, this is the third base. This is the first, second, third base. So CCU, the first letter is C. So if you come over here and look at the chart, the first letter is C, right? I said CCU. This is the first one. The second letter is also C. So the second letter here, if we come down, we have C, and we intersect with the letter C here, and the line was going to be U. So we see it right there. There's the third line. The third letter is along over here. Along, and there is CCU. First letter on this side, second letter up here, third letter over here. How about AAG? I'm just making these up. Okay, there's A here, so it's in this row. There's another A, there's the A for the second letter, that's in this, so it's in this block. And what did I say, AAU?
G.
AAG, so it comes down here, AAG is lysine. So you just, you use this chart. Carefully commit, remember, now you don't have to memorize it. Now you'll notice that a lot of them, you got for proline, for example, proline there on the second row and second column. Proline has 4 different codons, CCU, CCC, CCA, and CCD. So it can absorb a bunch of errors. If you accidentally code, you're making proline, and you accidentally code CCA, and you were given the instructions for CCC, it doesn't matter, it still works. Threonine has 4, alanine has 4, valine has 4, lysine has 2, asparagine has 2, arginine has 6 here and here. So you have a variety of codons for your amino acids. Up here is tryptophan. We've all heard of tryptophan, right? It gets blamed for making us sleepy after we eat a big meal of turkey or chicken. Not true, but it gets blamed for it. It really isn't true. Yes, so did I, but it really isn't. Tryptophan. How about phenylalanine? Oh, look, there's leucine. Leucine has 6 also right here. Phenylalanine. Anybody know what phenylalanine is? You ever use equal? It comes in a blue packet as an artificial sweetener. The active ingredient in phenylalanine is, in equal or NutraSweet is phenylalanine. So it's artificial. It's also often used as an artificial sweetener, but it is an important amino acid for us. So the genetic code, the triplet that we get from the DNA is converted to a codon on the messenger RNA that is the complement, and the messenger RNA codon specifies for these amino acids. So it doesn't matter what the codon is, we've got an amino acid for it here. So now the sequence of the amino acids in the protein is now spelled out by the sequence of the codons. So whatever the codon says is what's going to be the sequence of the amino acids. Proteins are made of amino acids. We link them together with peptide bonds. We can make them very long, 360 amino acids go into hemoglobin. They can be in the 20s, the 30s, it just depends. And since they can repeat themselves over and over again, it doesn't matter that we only have 20. Remember, 26 letters in the English alphabet, and we have 300 and some thousand words in a dictionary. So we can put any combination together to give us what we need. Now, you don't have to commit this to memory. So, but you do need to understand what the difference between the triplet and the codon is. The codon is just the copy of the DNA triplet. The codon is what we use to identify each unique amino acid. We have multiple copies. Again, leucine has 6, arginine has 6, valine and alanine have 4, threonine have 4 different combinations, proline and serine. Serine has 6, 4 there and 2 over on the far side. So we have lots of redundancy here, because mistakes on the order of the bases can happen. We're talking an error rate of 1 in a billion. Well, it's always possible to put a guanine in place of an adenine by accident. Well, it turns out in the case of proline, a guanine in place of adenine still works. If you make a mistake, though, what will happen is And if you can't fix the mistake. If the P53 can't fix the mistake, then that particular gene may shut down. But we got probably 10,000 more copies of that same gene. So if you make a mistake in one gene. You just, you let it go and it produces a non-functional protein, which is disintegrated by the body. Or you just shut down that segment of gene and cut that gene out. You know, a little one, you typically, if it's a harmless mistake, nothing's gonna happen. There's nothing, it's harmless, nothing's gonna go wrong. Transfer RNA, I know you're all waiting for this one. Bless you. Transfer RNA is a squiggly little paperclip shaped structure here with an amino acid attached to one end. And it's made, and like any RNA, it's made up of nucleotides. And down at the base here, we have what's known as the anticodon. The anticodon is also a triplet, but it's a triplet that is complementary to the codon. So if the codon was GGG, the anticodon would be C. That's going to be complementary to AUA. So this U is in place of thymine, so the complement to uracil is adenine. The complement to adenine is uracil. Complement to uracil is adenine. So that the anticodon is the complement to codon. So whatever you have in the anticodon, it will be the complement of the codon. So now we're going to bring our amino acids with these transfer RNAs are going to start bringing our amino acids to the messenger RNA. The messenger RNA is the sequence of codons. Sequence of codons in the messenger RNA determines the order of the amino acids. The transfer RNA will follow that order in bringing the amino acids to it in the ribosome. And it looks. So the process of translation starts at the beginning of the messenger RNA. There is a start codon and there's a stop codon. So our start codon is usually going to be AUA. Typically it's AUA, and the stop codon is and the anticodon to that is UAU. Let me show you what I'm talking about here. Okay, here we go. We have a start codon here that has AUG. Every protein starts with this amino acid called methionine. So whatever we have on our, if there's our codon, then our anticodon is going to be UAC. Go back here and see what's happening here. So translation doesn't make sense here in a second. We begin the process, we initiate the process, then it starts to grow and it elongates, and then we stop. So there is our ribosomal subunits coming together. Messenger RNA is surrounded by the two parts of the ribosome. It looks like a big hamburger bun. Okay, our very first, our start codon is always going to have AUG as its initiation point. The start codon always starts with AUG. And the complement to that, on the transfer RNA, the anticodon is going to be UAC, because the complement to A is U, the complement to A is U, complement to C is G. Everybody with me? So transfer RNA, the very first transfer RNA will bring in an amino acid that is unique to UAC, and that is methionine. Very first amino acid is methionine. The codon for methionine is AUG, and so we use the complement on the transfer RNA to bring it in. Back to this for one. AUG, methionine, that is the initiation codon. Every protein starts with methionine. And its codon is AUG. So now, here comes our ribosome. It has a small subunit and a large subunit. Transfer RNA lands on top of the codon. The anticodon and the codon bind briefly. And you've got your methionine, your very first amino acid, linked right there. Now the second amino acid is going to come in. On the second transfer RNA, it's going to come in and land in this spot. So the second one, the codon is GUA. The anticodon should be CAU. Because that's the complement, GUA, the complement to GUA is CAU. Complement to A is U, the complement to G is C, the complement to U is A. So we should get the second one coming in here. So, ignore this. Okay, here's our start codon right there. There's our ribosome forming. Here comes methionine, UAC, landing on top. Here we have the second codon is cytosine, guanine, cytosine. The second transfer RNA will come in that has the complementary set to that, which is guanine, cytosine, guanine. The two amino acids will link together with a peptide bond, and the first transfer RNA is then kicked out. The messenger RNA starts sliding through the ribosome and brings in a third transfer RNA that combines, that forms a pair, a complementary pair with the third codon. See it again. AUG comes the complement UAC. There's our methionine. There's the large and small parts of the ribosome. The second complex is cytosine, guanine, cytosine in the codon, guanine, cytosine, guanine on the anticodon. The amino acids bind. The first transfer RNA is kicked out. Here comes the third one. It's going to bond. GGA has a complement of CCU. We have another peptide bond, and the second transfer RNA is kicked out. And this process will continue building our protein chain until we hit what we call the stop codon. It will tell us that we're done. So the amino acids are brought in one at a time by the transfer RNA. The transfer RNA has the anticodon, which is the complement to the codon. And we'll continue to assemble until we hit our stop point. Here we see every time we have a peptide bond, a transfer RNA is kicked out. And this process will continue until we grow our protein to whatever size it's supposed to be. In this case, we're looking for a stop codon, and our stop codon here is UGA. Yes.
When the transfer RNA is kicked out, is it recycled or does it?
It's recycled back, pick up another amino acid. With the same codon. So, here we have ACU to CCA. So if this is going to get you methionine, well, that would work. Let me bring it back. Okay. So there is transfer RNA bringing in methionine right there. And when we kick the methionine, when the methionine attaches to the second amino acid, methionine, Now here comes the second amino acid. Bless you. And so you'll have a peptide bond forms there. This transfer RNA is kicked out. And it will go back and attach to another methionine. So. And so methionine is one of the uniqueness of methionine only has one codon for it. That's all that first transfer RNA will ever do. Now the rest of them can go back and select, you can have 6 different combinations, 6 different codons for serine. So you could have 6 different anticodon for serine. They're all gonna bring you the same amino acid. Or leucine will give you 6, or arginine will give you 6. But we take the transfer RNA and we cycle it back and it picks up its appropriate amino acid. Okay, now. So once we chug down along here, at some point, we're gonna hit the stop codon. One of the stop codons is UGA. There are 3 stop codons. If you're a fan of Jordan, you know what UGA means. So if you're not a fan of Jordan, you still know what it means. So UGA is one of the 3 stop codons. We go back here and look at this chart again. UGA, UAG, and UAA are all stop codons. So this is probably the one that's most well known of the 3. So we see UGA as a stop codon. Once we hit UGA, once we hit the stop codon, we stop. We have made our protein strand as long as it needs to be. Protein. And then that protein then is collected in a vesicle in the rough ER and taken to the Golgi apparatus, where it's joined together with all other proteins of its same type. And the Golgi will release it. So, yeah. And that's what this was going on here. There's your ribosome, the small unit, the large unit. That's not a worm coming out of an apple, even though it looks like it. The messenger RNA is chugging through. This is the messenger RNA in blue. The messenger RNA is going through the ribosome, and our polypeptide chain, the protein chain, is getting longer and longer and longer coming out of here. Then we hit the stop codon at some point, and we let go of the protein. The protein then is captured in a vesicle that is sent off to the Golgi. We put in the vesicle with all the other proteins just like it. The whole thing is based on the complementary pairing. And if you'll notice that the anticodon sequence of the transfer RNA is identical to the original DNA strand we started with. So the fact that we are actually using the DNA information, because we take the DNA, we make a complement of it on the messenger RNA. And so that's the opposite of the DNA. Now we make the anticodon on the transfer RNA, which is the opposite of the codon, which, if this isn't getting too convoluted, the anticodon is the exact sequence of the DNA. So we're back to the original DNA strand. And that tells us what we've got. Mutations can occur. We have what are known as substitution mutations. A mutation is any change in the order of a gene. Here we have CGA, ACC, CGA. Here we have CCA, CC, CGA. The substitution was right here. The original version of the DNA had a guanine as in the number 2 spot. Here we have a cytosine. Is that significant? It depends. We don't know. Well, actually, we do CCC or CGA. CCU. Oh, it won't do any, it won't have a problem because it's the same amino acid. So here was a substitution amino acid, substitution mutation. Make a difference. Substitution mutations can happen all the time and they won't hurt us. Because it's still coding for the same amino acid. It's when we code for a different amino acid, at that, in that codon, that we might have a problem. So. Hemoglobin, because I talk about this a lot, this is a good example of a simple little substitution error, genetically controlled. But caused by the substitution of the wrong amino acid. And it's caused by one base. Here we have a normal hemoglobin. In the number 6 spot, the number 6 protein spot, number 6 amino acid spot in the protein chain of hemoglobin, we have glutamic acid. In sickle cell disease, at the number 6 spot, we have valine. There's been a substitution of uracil for I'm sorry, of uracil here instead of adenine. Because look, GAG, the complement should be GAG, but it's not. We have uracils. And it only takes that substitution of one base to change the amino acid from glutamic acid to valine. Just one base. You know, the codon is GAG, here we have GUG. It's really, really close. But that substitution for uracil instead of adenine causes tremendous differences. It makes the hemoglobin brittle. It makes the hemoglobin curl up into the shape of a crescent or a sickle. It will cause the red cells to form a sickle shape and cause them to start blocking blood vessels and capillaries and cause great pain in the joints, particularly during exercise. And it reduces the oxygen carrying capacity of the hemoglobin by 80%. And this is an inherited disorder. It's not something that you acquire. One little mistake in the number 6 one, the DNA chain for hemoglobin is 360 proteins long. One little mistake in the number 6 spot, and it's only one base. It isn't even the codon. The codon is 66% correct. But we got the wrong base there instead. So the sequence can be very important.
So once they treat the anemia from sickle cell, then will does create or not stay to like treat it, or will it stay with that mutation?
Substituting recognition, or I bring them to the transplant, including LPD, so that it doesn't keep going treating it, transfusions and stuff. That's a band-aid.
Right.
It won't hurt. Place the subsequent, but they will still be in the carrier. You know, they may not be symptomatic, they still have the disease.
Still have to be taken.
Right.
Right.
And then sometimes, this is the last one. This will be on the test. I'm not gonna remember. Frame shift mutations. Here we cut out the wrong nucleotide. And then everything slides down one. So here's our original codon here, GCT, AGT, ATA. Now we cut out one. If we cut out this G and C, we cut, or we insert it. Here we're inserting the wrong set of nucleotides, but it can happen. Everything's pushed down wrong. We have a whole series of new codons, which are going to code, which either don't code correctly or code, you know, or they either don't code for a protein, or they start coding for totally different protein. Those can happen. Okay, and that is okay. Telomeres are compliant to last point. And we'll be done with this chapter and we'll start metabolism on Wednesday. Telomeres are probably the cause of aging. The more we chop away our telomeres, the more we age. The more rapidly we age. That's a theory. Okay, and that. Time to go. Thank you. Lab is.
Family and get crazy. Can't think about it now. Even my you know that they would care to. Right here. Oh, I'm perfect. I took it to when you're like this or things like things like
Hey, so I just have a question.
Sure.
What you mentioned there, but it could be crazy. I think that's on this